So as I was saying before we officially start, this is an intensive basic revision. So today I'm planned to finish all three chapters of genetic if possible, inheritance, variation, and genetic technology. This is just a basic revision program because for those to help those that are weak lah. If you're good or expert in the topic already, then it's okay because we won't be doing any like practice or what questions lah. Okay, okay. So let's begin. First of all, before you study Form 5, you you must understand how is the formation of chromosome in your cell first, which is studied in Form 4, Chapter 4. Okay, so let's have a look. Okay, so formation of chromosome from DNA protein. So your chromosome, they are basically the structure of the DNA in polynucleotide nucleotide chain that are worn around a protein called histone. Don't worry, I'll later draw on the whiteboard for you all. So histone do not carry genetic information, and then they combine to histone to form nucleosome and then intertwine form chromosome. So the chromosome structure is found in the nucleus of the cell lah. Okay, now, so first of all, you have to know first in the cell. Okay, in the cell you will have your nucleus and your nucleosome lah. Okay, this is a cell. Okay, this is the cell lah. Okay, so the cell, now we magnify the cell. You look in the nucleus, okay? We magnify it, okay? You will find that there will be a chromosome, okay? There will be chromosome. So the chromosome is like an X shape like that lah, okay? Then this is the centromere lah, okay? So chromosome, okay? Ini kita panggil centromere lah, okay? Is join the two sister chromatid. Actually, chromosome, it only exists like this form, you know, it's in the straight form. But like this will be when it's in the S-phase cell division, okay? Then it will be like this. Is the light refracting? The light. Okay, so centromere macam tu. Okay? Now, the short arm, we call it the P arm of the chromosome. The long arm, we call it the Q arm. Okay? So, this chromosome is actually contain a lot of DNA strands. A lot of DNA strands inside here, okay? A lot. And then you have your allele, some more which we'll touch later, okay? DNA strand. So, it's okay. We magnify again some more. We magnify some more, okay? So, you will find that there will be a lot of histone, protein called histone. And then the DNA will coil around this histone, okay? How do I do this? Okay, so all this is called a nucleosome. Okay, nucleosome, all right. Nucleosome will be in the chromosome compressed all together already. This protein is histone. Histone do not contain genetic information. Okay, do not contain genetic information. All the genetic information is stored in the DNA over here. Okay, the DNA, deoxyribonucleic acid lah, DNA, okay? So we have your genes, everything is in your DNA. All right, okay. And then finally, you magnify this some more, the nucleosome, and then you have your DNA lah, okay? Your DNA, and then of course you have your AGTC, ATGC, not AGTC. ATGC, whatever lah, ATGC. Understand? Okay, and then your DNA will consist of a double helix structure, right? It will be double stranded. So one side will be the 5, and then the other side will be the 3 lah. Okay, so basically this is it lah. So our genes all here. This is DNA. Okay. This is DNA. Okay. One section of a DNA, we call it a gene. The complete set of DNA, we call it a genome. Okay, so this is a gene lah. So from the gene here, the DNA, you coil around the histone, you get nucleosome. Compress everything together, you get the chromosome. Chromosome join at centromere. Then humans have you can say like, okay, 46 lah, 46 chromosome in the nucleus of one cell. Okay, one somatic cell. Okay, so this is the formation of our chromosome and DNA in our nucleus. Do you understand or not? If you understand this part, then only you can move to the next. All of you, do you understand? Okay, yes, but why the hell is it so complicated? Because humans, you have so much DNA. You only have 46 chromosome, but you have so much DNA, so you have to contain all the genetic information inside. Okay, 3N is what? Okay, that one actually isn't really in our syllabus, just an additional knowledge. Basically, that's like your DNA, you will have like a, I forget what is it called. Your polynucleotide, you will have like uh 3 5. Okay, I'll check back again what is it called, but basically the polynucleotide lah. There will be one N is 5 and one N is 3, but I'll inform you again. Okay, Eunice okay lah. Alia okay. Sabrina okay. Are you confused or you okay but actually you just think is complex but you okay lah? Okay, can lah. Okay. So maybe I can check now what is the 3 5 called. Hold on. But basically it's relating to the polynucleotide DNA 3 5. Okay. So okay. So one side is 5 prime, one side is 3 prime. The 5 prime and 3 prime refer to the number of carbon atom in a deoxyribose sugar. Okay. So just you last time you learned, you will have your you will have your pentose sugar. So here is your deoxyribose sugar lah. Okay, pentose deoxyribose sugar. You have your nitrogenous base. Okay, nitrogenous base. Okay, and then you will have your phosphate group lah. Okay, all this from 4 so must be in your head ready. Okay, so 5 prime means you have 5 carbon atom lah. 1 2 3 4 5. Okay, 3 primes mean you only have 3 carbon atom lah for the pentose sugar. Okay, understand? Yeah, but this one not your syllabus, so don't have to remember all this. Okay, I hope the words are clear. Is it clear? I hope it's clear like everything. Okay. If the number of buchu change, the name of sugar also change. Yeah, it depends, but in a DNA, you just focus on it's called deoxyribose sugar lah. Okay, understand? Pentose will become something. Okay, we just focus is called deoxyribose sugar lah. But basically, maksudnya dia akan bond dengan 5 lah. Okay, this is 5 prime and then there will be 3 prime. Okay, only 5 and 3 there won't be like 1, 2 or 4 like that. Okay. Okay, so I'll wrap everything already. Now let's see. Let's look at form 5. Okay. So inheritance. Who can tell me what is inheritance? Don't reply too slow. If you reply very slowly or you're waiting for others to reply, then we cannot finish the topic in time lah. What is the meaning of inheritance? What do you understand of inheritance when you hear this word? Inheritance. Similar traits and offering. Parents ada, you ada. Yeah, that's more like it. Other people. Okay, so basically inheritance, correct, is what your parents pass down to the offspring. Okay, so we will learn of 2 types of inheritance: monohybrid inheritance and dihybrid inheritance. Monohybrid means it will only inherit one trait. Okay, in reality, of course, we inherit so many traits lah, but now we will focus on one trait first. Okay, so now you look at the textbook definition of monohybrid cross. Monohybrid inheritance involves inheritance of one characteristic and contrasting traits controlled by gene. So that means you inherit the one characteristic or trait controlled by a single gene lah. Okay, okay, oh, okay. Many people told me, ayo, this Mendel experiment, very, why isn't it loading? Hold on. This Mendel experiment, they told me it's like very complicated, but actually it's not complicated lah. So it should be okay for you lah. Okay. So this Mendel basically is the father of inheritance because he invent or he discovered all these allele, genetics, everything, all this lah. Okay. So he did a P experiment to carry out, and then he found out all these traits and characteristics. Now, what is trait? What is characteristic? What is dominant? What is recessive? Later we'll have a look lah. So he found that the seed shape round is dominant, constrictor is recessive, seed color yellow dominant, green recessive, and all those lah. You can read yourself all here lah. Okay. So after that, after he carry out the experiment, he will draw a schematic diagram to determine what is the offspring characteristic. Okay. So in the experiment, Mendel used pure breed pea plant as the parental generation. Pure breed means the allele will be the same. Okay. Capital T, small t lah. Okay. Later I'll explain what is the meaning of all these pure breed lah, allele lah. Okay. Self-cross or pure breed tall plants produce only tall offsprings. Okay. Dominance principle. Okay. All this lah. Maybe later we'll come back to this again. Okay. Now we focus on the terms first. What are the terms related to inheritance? Okay. So the first thing is genes and alleles. Okay. So once again, pin my screen. Okay. Pin my screen. I'm going to draw on the whiteboard again. So we have to understand first what is genes and allele. Okay, so like I said just now, your DNA, I won't draw the ATGC, I'll just draw a simple one. This is your DNA, double helix structure. Okay, this is your DNA. Okay, so a single part of a DNA is called a gene, so they will control one characteristic. Okay, in this gene, it will control a single characteristic. But when we talk about allele normally we will already focus on the chromosome. Okay, so this is the chromosome lah. All of us have 23 pairs of chromosome. Okay, 23 pairs. So this is one pair of chromosome lah, one from your father, one from your mother. Okay, so allele it will be a single part of a gene focused on this chromosome. Let's say, for example, this is chromosome 21 lah, for example only, okay? Chromosome 21, okay. So this part of the chromosome, this special location will contain the genes of maybe your height, okay? For example, height. These are the alleles, okay? Alleles for maybe height, okay? On chromosome 21 lah, for example. Okay, so these alleles will always be located at the same position of the both pairs of the chromosome. Okay, so we call it the locus position. Okay, locus position. It will be located at the same locus. Dia tak akan satu allele kat sini, satu kat sini, satu kat sini. No. Okay, both will be on the same one. Maybe there can be another allele controlling maybe eye color. Okay, eye color. So both akan di sini lah. Dia tak akan satu sini, satu sini. Understand or not? This is allele. Okay? Okay? Others okay? Good. Okay, good. Okay, so you look at the definition. A gene is the basic unit of inheritance which consists of DNA segment lah. Okay, so DNA segment lah. Okay, control a specific characteristic lah. An allele is an alternative form of a gene for a specific trait located on the same locus of a pair of homologous chromosome. Yeah, because this is pair, so dia akan dipanggil homologous chromosome lah. Okay, one from your father, one from your mother. Okay, so an alternative form of gene is called allele. Okay, now what's the next term? Okay. Characteristic and trait. Okay, this one no need to draw lah. This one basically is saying, okay, characteristic and trait. Characteristic is a heritable feature such as height, eye color, blood group, and pressure. That means, apakah feature dia? That is characteristic. Tapi trait is the variation. Okay, in simple term. Contohnya. Contoh. Characteristic, I have height. Okay. Height ada apa? Tall. Short. Okay, so height is the characteristic. Tall, short akan jadi the trait. Eye color, for example. Eye color is characteristic. Trait can be what? Blue, brown, black, and so on. Understand? Yeah, dwarf. Yeah, green, whatever. Okay. Can you all suggest another characteristic and their traits? What else you all want to suggest? Okay, hair. Hair what? Hair type ke or hair what? Hair type. Okay, hair type. Okay, then what else? What trait will there be in hair type? Curly. Yes, good. Curly. What else? Straight. These two mean lah. Curly, straight. Okay. Gender. Yeah, gender also can. But gender, I won't really touch on. Call it a characteristic because gender is determined by your sex chromosome already. So we don't go until so far lah. Okay. 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 Phenotype and genotype. Now, phenotype is what you observe. For example, this height. Okay. Tall. Tall is the phenotype. Short is called a phenotype. You jangan confuse dengan trait lah. Trait means, yeah, dia trait. Tapi phenotype means like, for example, in terms of height, what is his phenotype? His phenotype is tall or short lah. Okay. You will only see what you can observe from the characteristic. Okay. Tall, short. He has brown eyes. He has black eyes. His phenotype is curly hair, straight hair. Understand? Tapi genotype means you have to relate to the gene already. Okay, genotype you have to tengok allele. Contohnya, tall. Kalau tall, okay, phenotype is tall, genotype mungkin capital T, small t, and capital T, capital T. Understand? Later we'll go in detail for this. Okay, this is genotype. Maksudnya, you will have your two alleles. Ini kita panggil genotype. Understand? Jangan confuse. Ini phenotype, ini genotype. Can you understand? Okay Can Okay, should be easy lah. Okay, what's the next one? Okay, so textbook you can go by yourself lah. Okay. Now, dominant allele and recessive allele. Okay, now this is the fun part lah. Okay. Dominant allele means the allele that is dominant lah. Dominant means like is the stronger one lah, expressing the trait. So the dominant allele will always express itself if that it is in the pair of allele. And we use capital letter to write dominant allele. But recessive, we use the small letter lah, okay? So dominant allele, dia akan suppress the recessive allele, okay? You see, the first one here, capital T, small t. Okay, capital T, small t, dia ada satu dominant. So, dia akan express the dominant trait here. Okay, kita akan abaikan the small one because dia akan suppress the characteristic of the small t. Okay, this one, 2 also dominant. Confirm lah, both dominant allele, that means dominant trait express lah, that means will be tall lah. But what if I have 2 small t? 2 small t, there's no dominant allele. So, only the recessive allele can be expressed lah. Understand or not? Okay. Now, this is related to dominant trait and recessive trait. Basically, if my dominant allele already has both capital T, so that means this is the dominant trait lah. The dominant trait is tall lah. Recessive trait will be short lah. Can you understand this part? Okay. I guess Jana, which one not sure? Which part? Is it trait not sure? Allele not sure? me sit down. This. Okay. You remember the bio exam, the chicken question. Ayo, go so far chicken already. Okay. Why the chicken? The comb kan? The comb, that one is the characteristic lah. Okay. Type of comb is characteristic. The trait will be the buttercup comb dengan strawberry comb lah. Yes, what's the problem? I won't go until schematic diagram that far yet. Hold on. Ratio you got lah. Oh, is it ready? I think one was two dominant, two was the heterozygous, one is homozygous recessive. And later we only go, okay, later we use the example later, okay? The last question I asked how you cannot dominant if both is recessive. Okay, that one I'll talk later. That one dia tanya parent tu, okay, dia heterozygous. So dia ada satu recessive, dia offspring dia akan satu. Nanti schematic diagram I akan ajar, okay? One by one, okay. Good. You asked the question. Okay, good. Okay. What's next? Okay, homozygous and heterozygous. Homozygous. What does it mean? Homo. What does homo mean, and what does hetero mean? Ayo. What does homo mean? Homo means same lah. Hetero means different lah. Okay. So homozygous means both allele are same. Capital or small. So ini homozygous. Ini pun homozygous. Okay. Capital T, homozygous dominant. Small t, homozygous recessive. Okay. Heterozygous, not same. One capital, one small. This is heterozygous. Understand? Dia confirm ada satu capital, satu small. Okay. Tapi kalau homozygous, you kena tengok homozygous homozygous dominant atau homozygous recessive. Okay. The next one is what lah? Okay, the next one is parental generation, filial generation. Okay, parental generation refer to first generation which are mated to produce. That means pure breed lah. Filial generation, okay. Pure breed refer to identical allele. Okay. Hybrid is the product of mating between two pure breed variety. Okay, so this one can combine lah. Okay, so basically, let's say your the parental generation, okay, the parental generation, okay, the allele dia akan confirm dia homozygous, dia akan sama punya. So capital T, small t, okay, dia confirm akan sama punya, pure breed lah. This is also what we call pure breed lah. Okay. When you cross them, later on we will learn that lah. When we cross them, okay. Okay. You will get this. You will get T. Okay. So this one, you see the alleles all mixed already. Pure breed is homozygous. Yes, correct. Alleles all mixed already. This is the offspring. So we call it the filial generation lah. You know, filial means what, right? Filial piety. Filial piety means in Chinese is 孝顺 lah. That means you respect your parents, all those. So filial generation is referring to the offspring lah, the child. So this will be hybrid already lah. Okay, you see all the different alleles mixed together already after fertilization. Okay, understand or not? Okay, so this is genotype or phenotype? Look at all this allele. This is genotype ke phenotype? Genotype, or geno. Okay, genotype. Now, what is the phenotype of this? What is phenotype of this? Tall. What is phenotype of this? Short. What is phenotype of all this? All this same, right? What is the phenotype of all this? Tall. tall, right? Because got the recessive, got the dominant allele, so it will suppress the recessive lah. You express the dominant trait lah. Okay. Okay, that's all. Next one. Okay. Both alleles are dominant, BB, therefore a dominant trait. Okay, so basically this is the purple color, white color, what I explained just now lah. The tall and the short lah. Okay, so you won't go into too much detail, that one. Ayo. Oh, no. Hold on, where is it? Let me reduce the size to 75 so you can see the whole page. Okay. Homologous chromosomes separate during meiosis. Okay. Okay. Second filial generation. This one you can read yourself. Okay. Now let's go to schematic diagram. Okay. Now schematic diagram is very important in inheritance. Okay. So let's use the example in exam. If you remember correctly lah. Okay, let's use the example in exam. Okay. We use the example in exam. Okay kan? Dia ada buttercup comb dengan strawberry comb. Okay. Dia ask you draw schematic diagram untuk kedua-dua dia adalah buttercup comb, tapi one was heterozygous and the other one yang anak tu heterozygous juga kan, if I'm not mistaken. Yeah, others, you remember? Okay, probably it's Winnie. Oh, Alia left already, is it? Forgot. Yeah, both buttercup. I remember it's heterozygous. Buttercup, we don't have homozygous because the question say heterozygous. Okay. Okay, actually I forget schematic diagram. At first, you have to draw the, what is it called first? You can draw the key first, sorry. Remember, you must have the key first for schematic diagram. You can express dulu. Okay, so key. BB means buttercup comb. Okay. Okay. Lepas tu, okay, BB strawberry comb. Okay. This once you already draw the key, okay, now you look. Okay, schematic diagram, you write the things on the left first. Why is it first? Okay. I thought, okay, if they tulis at depan, then I think it's not compulsory lah, that one. Okay, it's not compulsory. But let's say if they didn't lah, to be safe, always write more lah, okay, to be safe. But never mind, we will know lah next year how the schema. Okay, so you always remember this one. It will always start with parental phenotype, parental genotype. Okay, so you see, ini okay, I write short form lah, lazy write. Oh, okay, butter, okay, what? Strawberry, straw, okay, what? Okay, parental genotype BB. This one BB. Normally genotype we will use the rectangle to draw to indicate this is the genotype lah. Okay, parental genotype. Okay, lepas tu meiosis. Meiosis to produce apa? Gametes. Meiosis produce gametes. So gametes kita guna circle untuk draw. Okay, bring in the clothes. Ayo, okay, Nama, you can check the video recording later. Okay, so capital B, this one leaves were split during, because this is somatic cell, so haploid gamete you will split into two, capital B, small b, small b, small b. Okay, use the circle for gamete. They split 1, 2, 1, 2. Okay, do you understand until here? Okay, yes, okay. Now, after gamete macam mana? Fertilize. Okay, fertilization. And then you will form your filial Did they use filial or they use what? Okay, F1 genotype also can lah. Okay, okay, F1 genotype. Ayo, let me see. Oof, F1 genotype. Okay, so this one akan cross dengan ni. You see carefully. The first one cross with the third. The first one, you focus on the first one first. First cross with third, first cross with fourth. Never ever cross the same one. Because this is my own gamete, Sperm tak akan pergi fertilize sperm mah. Ovum tak akan fertilize ovum mah. So sperm dia akan fertilize ovum, sperm fertilize ovum. Okay? Now you done already with your first one, you move to the second sperm. Okay. This one fertilize with the third, this one fertilize with the fourth. Understand? So there will always be 4 lah. Okay? Because there will be 4 gametes mah. Okay? So 4 possibilities. Okay, then your genotype is what? B, capital B, small b. Capital B, small b. Okay, small b, small b, small b, small b. I mean, if this is correct. Oh, wait, I just remember something. Oh, no. I think I remember something. This one is supposed to be buttercup. I remember both is buttercup. Okay, this is supposed to be butter. Capital B. Okay, so dia akan menjadi B. Okay, B, 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 B. Okay, this one I think is, if I'm not mistaken. Okay, so then genotype. Okay, draw the rectangle. Okay, last one is F1 phenotype. Okay, then you write lah. Okay, this one, this is butter. Ini strawberry. Straw lah. Okay. If you want to do, you can also do ratio if you want. Okay. Okay, so you must have the 7 component: parental phenotype, parental genotype, meiosis, gametes, fertilization, F1 genotype, F1 phenotype. Understand? Okay, and then you can draw the ratio if you want lah. Okay, so from here you can analyze everything already. Both are buttercups, buttercup comb. The genotype is capital B, small b, capital B, small b. So maksudnya homozygous ke heterozygous? Hello, sudah tidur ke? Hetero, okay. Hetero, good, hetero. So you see, okay, dia akan produce satu ini. Dua-dua pun dominant. Ini homo ke hetero? Homo. Homo recessive or homo dominant? Dominant, okay. Ini hetero, ini homo recessive. So just now Jana asked, why both parents dominant trait butter? Tapi why dia akan produce strawberry recessive pula? Because of sini lah. Their parents both have the recessive gene. So when the recessive gene fertilize the recessive gene allele, dia akan jadi genotype dia akan recessive. So dia akan produce strawberry comb lah. Do you understand? So it's possible. Maybe both your parents have straight hair. But why suddenly you have curly hair? Because your parents have the recessive gene. So you also have recessive gene from both parents. So you akan express the recessive gene. Understand or not? Can you all understand the schematic diagram? Yes, good. Okay, if you understand this, that means your inheritance really okay. If you understand this, that means very good already. Who doesn't understand? Are you? Hey, Eunice, is she here? You takutnya. Okay, tak apa. We continue with the Punnett square. Okay, so another way we can express this one instead of drawing schematic diagram is we can use a Punnett square lah. But normally it's not in our syllabus. So the Punnett square is like this lah. Punnett square. Okay, so you draw lah. Okay, you draw. Okay, you akan draw 3 column and 3 row, 3 times 3 lah, so 9 boxes. So you put father punya wherever you want to put lah. Okay, capital B here, small b here. Mother punya capital B here, small b here. Okay. So, cross dia. B dengan B, capital B, both. B, capital B, small b here. Small b, capital B here. Small b here, here. So, you know lah, this is 3 to 1. Can you understand? This one got in textbook, but normally exam, they won't test you lah, okay? Do you understand this? So, you see, from all this very complicated, we simplify into this only. Do you understand? Do you understand how to draw it? Minum air dulu. Sabrina? Can lah, okay. Okay, we continue lah. So hopefully Eunice watches the recording. Okay, Mendel's first law. Okay, so basically after this, you know already lah what is Mendel's first law. Law of segregation, which means a characteristic of an organism is controlled by a pair of allele. Only one pair of allele is inherited in the gamete. That means, okay, let's say for this one, this characteristic, the comb type of comb is controlled by this allele lah, B allele. Dia tak akan dikontrol oleh A lah, Z lah, X lah, no. Only this B allele akan control the type of comb. Understand? And dia gamete, dia hanya akan ada satu gamete, haploid hanya ada satu allele saja. Understand? Because you remember, just now your homologous chromosome, dia akan split mah. Ini satu gamete, ini satu gamete. So sini ialah satu allele, sini satu allele. Do you understand? Yeah, okay. So, what are the other specifications of Mendel's first law? Characteristic, inheritance, next generation, genetic factor known as gene. Trait is controlled by a genetic factor, no allele, okay lah, blah, blah, blah. Okay, so why is it called law of segregation? Because dia akan segregate. Dia akan meiosis, dia segregate, dia separate. Okay, okay, what else? During fertilization, okay, possess two allele, fertilization is random, so okay. Okay lah, so that's all for monohybrid. Dihybrid is actually easy. It's just that it becomes complicated because now you have two alleles. Okay, two types of allele. So dihybrid inheritance involves the inheritance of two characteristics. Each characteristic is controlled by a different gene located at different locus. Okay, you see, humans, we have billions and billions of characteristics. Takkan kita satu gamete only have one allele? So now kita akan ada multiple, a lot of alleles inside. But in our syllabus, we just learn mono and di only, okay? So 1 and 2 only, okay? Now, the same thing as mono, except that we change to di lah, that means two characteristics lah. Okay, so, okay, how should I do this? Okay, let us change the schematic. We follow textbook lah, okay? Okay. Okay. Let us change the schematic diagram to dihybrid. Okay, so what is this now? Okay, BBKK. Okay, let's say I have a homozygous dominant BBKK. Okay, you see two different letters, maksudnya dia akan two different characteristic dah. Okay, BBKK. Okay, so round and yellow. Okay, round yellow. Round yellow. Okay, let's say lah B2 akan control the round, K is control yellow. Okay. Okay, now then the next one is the recessive, right? Okay, constricted and green, BBKK. BBKK. Constricted, green. Okay. Now, parental phenotype. Okay, we just copy here. Copy here only. Okay. I malas tulis already. Okay, BBKK. A, B, B, K, K. Okay. Now, parental, okay, this is parental genotype. Meiosis divide two again. Actually, your meiosis, dia boleh banyak. Dia akan produce dua, it will only produce two, but actually you have four possibilities lah, okay? Nama we draw four. Okay, four possibilities. So here you see like I said just now, one gamete dia hanya akan ada satu allele saja. Dia tak boleh both the same allele. So sini you pilih satu lah. Okay, capital B. Sini you pilih satu lagi, capital K. One gamete. Okay. So if you, let's say you want to draw it like this, you see satu gamete dia akan ada satu chromosome. Sini maybe is B, sini K. Ini B, ini K. Understand? Different locus, different allele at different locus controlling different characteristics. Okay? BK, this one, capital B, capital K. Capital B, capital K, capital B, capital K. Okay. This one, small b and small k. Same thing lah, small b, small k, small b, small k, small b, small k, small b, small k. Okay. And then you fertilize lah, okay? We don't do so many because this is so complicated. So you just do one. Okay, let's say this one. So they akan produce B, B, K, K. Capital B, small b, capital K, small k. Okay, B, B, K, K. Okay. Now, tell me what is the phenotype? You look at the color first. Round, the shape first, texture first. Round or constricted? Round. Oh, hello Eunice, you're back. Round because ada capital. Dominant to round. Okay. Color? Yellow. Good, because dominant. Okay, good. So if you use the Punnett square is more complicated already, as you see in textbook, because you have four types already. So dia akan 4 times 4 lah, become 16 already lah. Okay, I won't draw it here lah. Okay, it's just the same thing. Do you understand? This is dihybrid lah. Basically, you just add on one more allele inside. Can you understand? If you understand this, I give you 10 allele, so you can do already one actually. It's just the same concept. So Mendel's second law, independent assortment. During gamete formation, each allele from a pair of allele can combine randomly with any allele from another pair of allele. New combination of characteristics are produced in the F2 generation. Okay. Characteristics are controlled. React freely. Okay. I cannot scroll down. Okay. Now we go to the third subtopic. Actually, this one also I already touched like just now. Basically, you see this homologous chromosome, dia ada allele, dia ada so each dia homologous punya chromosome, dia akan ada satu allele at the same position, the locus position. Okay, the A beside is also A, C, C, F, F, J, J, M, M. You understand? You understand this diagram? The one I'm sharing in the PDF one. Can Okay. Yeah, same matter only, Eunice. Okay. Okay, now you look at inheritance, two types of human chromosome, autosome and sex chromosome. Okay. So humans, okay, the gametes, akan ada. Okay, actually we don't focus on gamete, we focus on normal human first. Human being, we have 46 chromosomes. Okay, among these 46, we have 44 autosome, 2 sex chromosome. Okay, the 2 sex chromosome itu akan jadi XX atau XY. Okay, XX is girl, female, XY is boy. Okay, So maksudnya 44 dia akan ada 22 pairs of autosome and then one pair of sex chromosome. Okay, so you look at the, what is it called already? Yeah, this is gone. Ayo, okay, tak apa, kita continue. You look at the karyotype. Okay, you look at the textbook punya karyotype. So situ dia ada 22 pairs of autosome and one pair of sex chromosome. So XX, XY. So you notice X is longer than Y lah. Okay, because Y is normally cannot express punya. Okay, so when I want to produce a gamete, as I said, is haploid. So it will always be half. Okay, half. So half itu jadi 22 and then sini half lagi. Depends lah. Okay, girls you can only produce ovum with X. You cannot produce Y punya ovum. You can only produce X. So 22 plus X. Boys, you can produce 22 plus X punya sperm atau Y punya sperm. Itu possible. Or 22 plus Y. Understand? So the boy punya sperm, you akan determine the gender of the child lah. Okay? So 22 plus fertilize with 22, 44, and then X fertilize with X, you get your sex chromosome, so 46. Do you understand this? This is humans lah, very, very important but also basic. Can you understand? Yeah, very easy only, right? Jana, Winnie can write, I assume. Okay. Okay, apa ni? Okay. Let's say you see the, okay, you look at the textbook. Okay, you see suddenly total chromosome number here is 47. What happened? Normally 46 tapi sini 47. Then you look at chromosome 21. Supposedly dia akan satu pair tapi dia ada 3. So kita panggil ini trisomy 21. Okay, trisomy means 3 lah. So this syndrome is called Down syndrome. This is a chromosomal mutation. We will learn more of this in the mutation in the upcoming chapter lah. Okay, mutation. So you see, it will occur in both males and female because it's only at the autosome. 1 until 22 is autosome, bukan sex chromosome. So dia akan occur dalam male and female lah. Okay, Turner syndrome macam mana? Turner syndrome, dia akan ada 44 autosome, cantik, tapi sex chromosome dia hilang satu. Dia akan hanya ada satu X. Dia X and O. Dia hilang satu sex chromosome. Itu Turner syndrome. So it occurs in female lah. Okay. Klinefelter syndrome, autosome cantik, tapi sex chromosome dia extra satu X. So this occurs in male. Klinefelter syndrome. Okay. So dia akan extra satu X. Do you understand all this? This one, if later on you also mutation, we will look at it. Can you understand it? Can. Okay. Good. Now we look at inheritance. ABO blood group. Okay. Now, I want to ask you all, what are your blood groups? Jana, what's your blood group? I'm O positive. Darwina, Jana, Winnie, what's your blood group? A positive, A positive. Ooh, okay. Wait, semua A positive. Oh. Sure, it's not because SPM you want A plus, is it? Okay, A positive lah. Okay, A positive. So that means, okay, now we'll see what does it mean lah. Okay. So blood group, the alleles is a bit special because you won't have like you won't have just now those like A, B, B like that. You will use I as the base first. You akan guna I as the base. You akan ada 3 jenis allele. Because blood group is interesting. You have 4 types of blood group. You akan ada 3 types of allele. You have I, A, I, B, I, O. Okay? So, let's see. A and B. See this. Okay. A and B, dominant. IO, recessive. Understand? So, in simple terms lah, A tu, dia akan jadi, if let's say my blood group is A, which is all of you lah, the 3 of you, blood group A, it's possible your allele is IA, or IA, IO. Okay.
IA, homozygous or heterozygous? Homo dominant, homo atau homo recessive? Dominant, okay. IAIO, hetero or homo? Hetero, okay. So if you are A type, then your blood group can be IA or IAIO lah. Okay. Eunice, what's your blood type? A. Oh, wait, semua A. Okay. Okay, tak apa. Okay, now B. Look at B. If your blood group is B, same thing lah. You have the dominant IB and you can have the homozygous dominant or IBIO. O is recessive, so dia tak akan express O. Okay. Kalau AB, blood group, one thing interesting about blood group is dia boleh ada dua dominant different pair of allele. Kita panggil dia co-dominant. Okay. IAIB, co-dominant. Maksudnya both dominant, dia pun sangat proud. They want to express. A mau express dia, B mau express dia. So both co-dominant express each other. Understand? O people going extinct as we speak, but O is the universal donor. So you know O, then you all die lah. Okay. So yeah, this is AB. So IA, IB, co-dominant. Okay. Understand? All of you? The AB group? A. Anti-A. AB universal receiver do better. No lah, it's better to have donor. What? You other things to how to say? You have your supply ma. If you have banyak receiver but no one to give to donate, then you akan extinct what? Okay. Co-dominant is both also dominant. Yes, and this is only in blood group. Okay, blood group only akan co-dominant. Okay, because we have 3 allele. Better for mother nature. Okay, the last one O. So saya lah, saya yang paling kasihan lah. Okay, kesian lah. So O, I'm only recessive lah. Both of my alleles are recessive lah. IO. Okay, so this is homozygous recessive. Okay. Now we look at the inheritance. Okay. Satu father ada IAIO. Satu mother. Okay, never mind. We make this more interesting. How should I say? Okay, never mind. My father blood type is O. My mother blood type is B, if I'm not mistaken. B. Okay. We assume this is O lah, okay? Because I have to, for me to get the O, I need to have the O from my mother ma, okay? So kita guna me as example, myself, okay? My father is IO, my mother is IBIO, okay? So my mother, her blood group is B lah, my father blood group is O lah, okay? But so how meiosis again? IO, okay. IB, IO, IB, and then IO. Okay. Now, fertilization, IO dengan IB. So, IB, IO, okay. IO. Okay, IBIO, IO. Okay. So this will be the B blood group lah. This is A blood group. That's why my sister is B blood group. Me and my brother A pula O. My brother and I are O lah, but my sister is B lah. Understand? So dia tak akan ada AB lah. Dia tak akan ada A. Dia tak akan ada AB. If your parents, one is IO, one is B, and then suddenly got someone keluar, the child is AB or A, that means something wrong already lah. Okay, so your child only can B and O lah. Understand? Okay, yeah, maybe a doctor or even worse lah. Okay. So this is just the blood group inheritance. Okay. Rhesus factor. Okay, rhesus factor actually is the same thing. It's just that rhesus factor it will become because of the antigen D. So dominant is Rh plus, recessive is Rh minus. So you see the same thing lah, rhesus plus, rhesus minus. This one I don't have to draw out lah. It's just that you see lah. If it's both, if it's Rh plus dominant, that means rhesus factor positive. Rh plus, Rh minus also rhesus factor positive, but Rh minus, that means negative lah. Okay. Okay, thalassemia. How do I do this on an autosome? Okay, thalassemia, the gene is controlled on the chromosome 11 or 16. Okay, chromosome 11 or 16. How do I do this? Mother normal, father carrier. Let me think about this first. Sex linked inheritance. Okay. Carrier maksudnya dia heterozygous lah. Okay. Thalassemia dia akan jadi heterozygous. Tapi okay, let's try. I got confused with minor thalassemia. Thalassemia minor. Okay. Jom kita buat. Okay, jom kita buat thalassemia. Okay, pilih satu alphabet. What you all want? Except T lah. T is already hard. Everyone think is hard already. Pilih satu alphabet. A. Okay, let's go A. Thalassemia. Okay, T again. Okay, A. What is the example? A normal. Okay, kita buat carrier lah. Okay, carrier. A. Okay. A is thalassemia minor. Because thalassemia is a recessive allele, okay? Recessive punya karakteristik, recessive punya. Okay? Kalau capital A normal. Kalau A both small, because both recessive, so that means confirm lah thalassemia major already lah. You kena thalassemia already. Okay. Now, okay, father, let's say minor, thalassemia minor. Thalassemia minor means carrier only. You only have the one recessive allele, but it's not expressed because your dominant punya normal you suppress. Okay. Okay, and then thalassemia minor mother. Okay, again, meiosis dulu. Okay, fertilization. Okay, A normal. A minor. A minor. A. Thalassemia major. Okay, so normal 50% will be thalassemia minor. 25% your child will be thalassemia major lah. So if let's say you have thalassemia minor, you kena hati-hati lah. If your partner has thalassemia minor as well, or even worse major, that means your child confirm got the risk lah of having thalassemia. Okay, understand or not? This is how it works lah. Thalassemia is the recessive allele lah. Do you understand? Yes, Okay. So this is thalassemia. Sex determination, I think don't have to explain because just now I explained already. Sex-linked inheritance basically is like color blindness, hemophilia. All this is because of the sex chromosome. Dia bukan di autosome, dia di sex chromosome. Okay, I just draw one example. Okay, let's use color blindness lah. Okay. Okay, all these sickness you kena ingat dia semua recessive semua akan recessive. No sickness is dominant punya. Okay. Semua recessive. Okay, let's say color blindness. Okay, because you see, this is already a sex-linked inheritance disease, so you kena tulis XY as the base first, X and Y. You cannot use AA or this. This thalassemia is autosomal saja. Okay. Color blindness. Okay, T. This is why it gets a bit complicated. Okay, boy kita ada 3 jenis. Boy ada 2 jenis saja. Girl akan ada 3 jenis. Okay. Remember, just now I said why dia short. Why itu actually is no use one. Dia tak ada gene. So, semua akan dikontrol oleh X untuk boy. Okay, ini boy ya. Ini girl. Okay, boy, girl. Okay, so boy, if let's say capital letter, okay, pick one letter. Okay, let's go with C. Okay, C. Capital C, boy tu normal. Capital mah, dominant is normal mah. Okay. Small c, you ignore the Y because the Y dia tak ada special ability. Dia tak ada gene punya. You just look at the X. So, boy dia akan ada color blindness lah. Recessive C. The boy, there is never color blindness carrier punya boy. Understand? Because only the X can control. You look at the table, male carrier tak ada, doesn't exist. Okay? So boy, you are either normal or you memang carrier lah. You memang kena lah. Girl, there are 3 types. Normal, X capital C, X capital C. Carrier, capital C, small c. You ada satu recessive allele. Okay. You kena color blindness small c, small c. Okay. Okay. Now, what example you want to choose? What the text for example? Normal eyesight carrier. Okay. Normal dengan carrier. Okay. X capital C, Y. X capital C, X small c. Okay. Okay. Tengok meiosis. Okay. My sperm normal, Y sperm, ovum normal, ovum with color blindness allele, fertilization. Okay, XC both capital. This is normal girl. This one dengan ni carrier girl. Should call this smaller bigger. Carrier girl. This one and this normal boy. This one and this boy yang ada color blindness. Do you understand? Yeah, you see, boys and girls is always 50% boy, 50% girl punya. Can you understand? So, normal father, carrier mother, 25% normal girl, 25% normal boy, 25% is carrier girl, 25% is boy yang kena color blindness. Can you understand? So the same thing is for hemophilia. Same thing saja. Only the same thing. Okay? Others, do you understand or not? Okay. Sabrina. Okay. Jana. Okay. Okay, good. Hemophilia. Okay. Ability rotang is the same. Okay. Family pedigree. This one not too important. It's just basically showing the characteristic and inheritance lah. So let's say you have a disease. Let's say you personally have thalassemia or whatever, then you can trace back, maybe you ask your mother or father or your grandparents, do they have thalassemia? And then you can trace back lah, understand, to the ancestral tree lah. And then you see lah who is the one responsible for causing your descendants to become carrying thalassemia. Okay, so this is family pedigree lah. Okay, habis chapter 11. Now chapter 12 is variation. So now let's go to variation. Basically, yes, variation is the difference in characteristic lah. He is tall, he can be short, all these different variety lah. Okay, so variation, we have two types. Okay, first we have to look at the necessity of variation first. Okay, enables natural environment to continue selecting beneficial characteristic and remove non-suitable one. That means you have variation so that you can the natural environment will choose who is the most suitable to live to ensure the continuity of the species lah. We call this natural selection. Okay, ensure survival of the species when the environment change, allows crossbreeding among species to form new species. Because if there's no variation, let's say you and your partner, your wife and your husband, 100% the gene same, then your offspring confirm akan mati lah because your gene is so weak, there's no variation. 100% the same, there's no variation, so your species akan mati lah. That's why we need variation, understand, to create strong genes. Okay. Now, types of variation. We have continuous and we have discontinuous. Okay. Continuous. Discontinuous. Okay. Now, what is continuous? What is discontinuous? You have to know first. Continuous variation maksudnya difference tu dia tak distinct, dia boleh di measure. Contohnya dia satu spectrum. Okay, I think the graph, I draw graph dulu dia lebih senang. So shape of the graph untuk continuous, dia akan bell curve macam tu. Dia akan continuous saja. I continue macam tu. Tapi discontinuous, dia hanya akan satu macam tu, satu macam tu, satu macam tu, satu macam tu. Understand? Different punya. Dia akan ada space macam tu. Tapi continuous dia akan continue ni. Continue, no break like that. Understand? Okay. So, because of this, continuous variation, kita panggil dia quantitative. Untuk sini, quantitative. Quantitative. Untuk discontinuous, qualitative. Quantitative maksudnya you boleh measure. Contohnya, you pick the wrong graph. Ayo. Contohnya here you see, 150 cm my height. Here maybe 160, 170, 180. You can continue on 151, 152, 153, 154. So, semua continuous macam tu. That is for height lah, which is an example of continuous variation. Okay? This continuous, dia tengok quality saja. You cannot see the difference. Dia bukan height or what. You only can see contohnya macam apa. Okay, what should we do? Okay, blood group lah. Okay. A, B, O. I tak boleh measure. I tahu saja the type A, B, O. Macam tu saja. Can you understand this part? Okay, can I? Okay. Now, the another properties of it is that it is influenced by environment. Influenced by environment. This one, not influenced by environment. Maksudnya, your lifestyle, for continuous, your lifestyle, your whatever environment can affect. If let's say you play basketball a lot, you very sportive, you or yeah, maybe you eat healthy food, then of course you are taller lah. Okay, but if let's say you don't like sports, you eat unhealthy food, then you'll be shorter lah. This is affected by the environment. But here, discontinuous, dia tak boleh affected. I blood group A, macam mana my lifestyle, I will still always be blood group A until I die. Blood group O also same. All these blood group also macam tu. Understand? discrimination to short people. Yeah lah, but I'm short also lah. Sabrina got call, okay, tak apa. Okay, so the property. So now we look at example. Example, apa yang akan boleh measure? Height lah. Height, mass, apa lagi? Skin color, because the tone. Skin color pun boleh. Okay. Weight, yeah, weight lah, mass lah. Okay, good. Okay, discontinuous variation. Example, you got blood group. Apa lagi? Type of earlobe. Basically, you see type of what? Type of what is discontinuous lah. Type of earlobe. Okay, eye color. Ability to roll tongue. Okay. Understand? Number of leaves is continuous. Yes. Number of leaves is continuous. Because you can measure quantitative except maybe shape of leaf, itu qualitative. Discontinuous. Shape of leaf. Okay. So, okay, the difference and the similarity and differences, I think you also know already. You can read textbook later on. Causes of variation. Actually, just now what I want to say. Hold on. Causes, environmental factor. Okay. I skip to this first. You have to know which one is dominant, which one is recessive first. So, height. Tall is dominant, short recessive, type of hair curly dominant, straight recessive, right-handed dominant, left recessive. Dominant dimples present is dominant, dimples absent is recessive. Can roll tongue dominant, cannot roll tongue recessive. Free earlobe dominant, attached earlobe recessive. Okay, let's say you use me as an example. I'm short, so I have recessive allele. I have straight hair, I have recessive allele. I am right-handed, I have the dominant allele. I don't have dimples, I have recessive allele. I cannot roll my tongue, I have recessive allele. My earlobe is free earlobe, so I have dominant allele. So you see, one person, your alleles and traits can be different from each other. Maybe this one I dominant, tapi untuk ini I recessive. Ini dominant, ini recessive. You understand? Okay. Okay, now we 12.20 mutation now. Okay, now we go back sini sikit. Actually, this one is related to that one lah. Okay, related to cell division. Okay, genetic factor, dia akan affect variation, environmental factor juga. Okay, genetic factor. Remember, genetic factor there will be the three main one during cell division, and then mutation is yang how to say, yang akan produce the pelik one lah. Is not normal lah, is abnormal. Okay, mutation. So the first one you see, crossing over. Crossing over occur between non-sister chromatic of the diagram is actually very good, so I don't have to redraw again. So non-sister chromatic during prophase 1 of meiosis. I hope you remember, okay, your cell division. So because you cross over, exchange of genetic information, maksudnya your offspring akan berbeza gene already lah. You don't look the same as your father and your mother, right? Because you have your exchange of genetic material already during prophase 1 lah. Crossing over at where? Between non-sister chromatic at where? Which part again? Revision. Crossing over at yalah, the stage is prophase, but which part of the chromosome? Chiasma, yes. You can say, chiasma, okay? Crossing over at chiasma. Okay. The second factor is independent assortment. What does it mean, independent assortment? Maksudnya, your cara you arrange the chromosome. You look at arrangement 1. The green on the left, the purple on the right. Okay, this is one type of arrangement, one possibility. So the chromosome, the gamete akan macam tu lah. Tapi arrangement tu you see, green left, purple right, purple left, purple green right, and then different arrangement again. So this will also produce different type of gametes lah. So this is called independent assortment. Chromosomes homologous chromosomes are arranged independently and randomly at the equatorial plane. Do you understand this? Independent assortment should be okay, right? Okay, random fertilization basically means like billions of sperm, one ovum, so many different types of sperm can fertilize the ovum. So depends on which sperm. You understand? Random fertilization is more towards logic lah also. It's not really scientific lah. You don't have to study bio to know that. Okay. The fourth one is mutation. We will look into later. Mutation creates new genotypes. Okay. So mutation, there are two types: chromosomal mutation and gene mutation. Later we'll look at it lah. Okay. Environmental factor. Okay. Light and pH, temperature, all this lah. So this one you can read on your own lah. Okay. Environmental factor. Okay. This is why I say like interaction between genetic and environmental factor. Like you see, J and K identical twins, but are they the same? Do they have will they always have the same height, same weight, same skin color, same this and that? No, because their continuous variation can be affected by the environment. Let's say you see J and K were separated since childhood. J eat healthy, tapi K tak healthy. So K obese lah, J ideal body weight lah. Understand? Okay, regret swimming to that oval now. Okay. Okay, variation. Now we look at mutation. Okay, mutation ni sangat seronok lah. Okay, menarik lah. Okay, mutation. A substance that causes the mutation is called a mutagen. Okay, and then the genetic material produced by the mutation is called mutant. So the mutant dia boleh because of the gene, dia boleh because of the cell, dia boleh organelle, dia boleh individual, dia boleh organ, okay? Dia boleh tissue, okay? So mutagen, you have three types. One is physical, one is chemical, one is biological. Okay. Physical agent, basically your UV ray or whatever radiation lah, okay? Physical agent. Your radiation will affect the cell division of your cell and the growth of your cell lah. So it will cause mutation. Chemical agent, let's say carcinogen in your cigarette smoke, or your food preservative, formaldehyde, benzene, if you're exposed too long, then also can cause mutation. Biological is virus and bacteria lah. Okay, will cause the mutation, especially during pregnancy. Okay, so types of mutation. Ini yang paling seronok. Gene mutation dan chromosomal mutation. Okay, siapa boleh explain dulu apa tu difference? Gene mutation tu apa? Chromosomal mutation tu apa? It's okay. Okay, mutation, you have two types. You have gene mutation and chromosomal mutation. Okay, gene mutation basically dia akan affect your DNA sequence. Dia affect the gene or your DNA sequence. Okay, actually, this is one of the part like I scared to talk about because it's very heavy. Yeah, it's easy, but then online very hard to teach because this one involves transcription and translation. You know, it's gonna be so hard to explain. I'll try my best, and I'll try my best not to go too deep into it. Okay. Chromosomal mutation, on the other hand, is affect the chromosome, number of chromosome. Bukan the gene, dia chromosome. So maksudnya dia tak akan 4, 6 macam tu. Something will confirm go wrong here. Dia tak akan 46 chromosome. This is chromosomal mutation. Gene mutation affect gene or DNA sequence. Okay. Now we look at gene mutation first. What is the cause? Okay. Now gene mutation is also known as point mutation. Okay, so they are the tiga: base substitution, base deletion, base insertion. Okay. So you see. How do I do this? Okay, there are three types. Base substitution. Now you look at the first example. Base substitution. Okay, dia T tu dia tambah satu, bukan tambah, sorry. Substitute, that means you exchange. So T tu tukar dengan G. So dia produce G. Okay. So base insertion macam tu. Okay, insertion maksudnya dia akan tambah lah. Deletion maksud dia delete satu lah. Okay. Do you want me to explain about transcription and translation? Production of the amino acid in your cell. Do you want me to explain that? Let's go with it lah. Okay. Now, how does your cell produce protein? Protein is amino acid. So you have to produce the amino acid. Where is it synthesized? Which organelle synthesizes amino acid? Ribosome. Good, Eunice. Ribosome akan synthesize the what is it called? The amino acid. Lepas tu rough endoplasmic reticulum akan transport lah. Okay. Lepas tu Golgi apparatus process ke whatever. Okay. So there are dua stages. One is transcription, one is translation. Transcription. And then you have translation. Okay, transcription occurs in the ribosome. Oh, I mean, it can also be in the wait, sorry, it's not the ribosome. Ayo, salah dah. Sorry, it's in the nucleus or nucleosome lah to be exact. Okay. Okay. What happens is our DNA. This is our DNA. Okay. DNA double helix. Okay. Double helix. Okay, so the DNA dia akan produce satu mRNA. Dia akan transcript satu polynucleotide mRNA keluar. Okay, so let's say okay, DNA, what base do we have? What nitrogenous base do we have in DNA, and what nitrogenous base do we have in the what is it called? In RNA. RNA is only one strand so dia akan satu strand saja macam ni. ATGC, okay, ATGC akan untuk DNA. Untuk RNA? Uracil, okay, uracil dengan siapa? Tukar dengan siapa? T, yes, correct. So, DNA, ATGC, RNA, AUGC. Okay, let's say A. ATGC, CGT. Okay. mRNA akan produce apa? Dia tak akan pairing. Okay, tengok. DNA A, mRNA sebenarnya A akan pair dengan T. Tapi mRNA dia tak ada T. So, dia akan ada uracil, U. T dengan siapa? A. G dengan C, C dengan G. Do you understand this? Yes, good. Okay. C dengan siapa? G. G dengan siapa? C. T dengan U. Sorry, T dengan A. Sorry. T dengan A. Okay. So, the mRNA ini akan jadi the sequence code. Amino acid punya sequence code. Okay. Lepas tu, translation akan di ribosome to produce the amino acid. Look at the whiteboard Okay, translation. Let's say I take this. 3 akan jadi satu codon, 3 satu codon. This will become one codon. This will become one codon. To produce one amino acid, you need 3 codon. Not 4, not 2, not 1, 3. Okay. So, translation di ribosome, mRNA akan pergi situ. Okay, we copy the same thing. U, A, C, satu kodon. G, C, satu kodon. And maybe, okay, A, U, okay, A, U, G lah, okay, for example. Okay. Dia akan produce satu amino acid, satu amino acid, satu amino acid. Okay. Now, I will share this. So this amino acid table will determine what is the amino acid produced. You see, first nucleotide, for this one UAC, first nucleotide you tengoklah U, A dengan C. So dia akan produce tyrosine. Okay? This one draw too small. Dia akan produce tyrosine. What is that? Tyrosine. GGC akan produce glycine. Okay. AUG akan produce methionine. So this is how lah. Okay, habis. Understand? This is translation, then your amino acid produced ready. Do you understand? Okay, you pin my screen. You pin back to my screen. Do you understand first? Yes. Jana, confused already. No need memorize amino acid. No need. Not your level, no need. Anyway, in the upper level, they will give the table already. So tak apa. Okay. Understand or not? Sabrina, I think you cannot ready, is it? Jana also cannot ready, is it? Eunice can no. Surprisingly, Eunice can. Very good, eh? Okay, Jana. Okay. Okay. Now, gene mutation happens when suddenly one of these goes wrong. Kita buat substitution lah, senang. Okay, substitution. Let's say UATG, UAC. Okay, UAG. No, I don't like that. CCC. So glycine. I'm saying glycine. Hold on, let me think of a sec. Think for a second. GCG. Okay. Let's say, you tengok sini, pin my screen. CCG. Let's say this C suddenly I substitute become G. Suddenly something wrong. G sini. Kalau sini G, sini akan apa? mRNA akan apa? C. Good. mRNA C. When mRNA C, look at the amino acid table, GCC akan produce apa? Dia bukan produce glycine dah. Dia akan produce GCC akan produce alanine. Dia akan produce alanine. So, protein dia, the code dia, sequence sudah salah lah. Sepatutnya sini glycine, tapi dia tukar kepada alanine. So, this is gene mutation lah. It affect, the gene here affect already. So, when the gene affect, sini akan affect. The protein amino acid akan affect. Do you understand? We don't have to identify what they produce, right? Yeah, we just, you don't know, you don't have to memorize the amino acid. You just have to know ini gene mutation. Mutation at here, the gene dia tukar. That's why the amino acid dia akan tukar. Do you understand? Good. Okay, good. Okay, the most important, Sabrina and Jana, if you don't understand, is just that you know gene mutation akan affect gene dia. That's why production of protein akan affected. Understand? Spider-Man gene has mutated after. Yes, correct. Maybe the protein the protein something wrong already here. That's why superpower. Okay. So in substitution, insertion, deletion okay, you have to know this, and then you have to know example. Okay, sickle cell anemia, cystic fibrosis, thalassemia. Okay, albinism. Hold on. Okay, this one I erase already. Okay, good. it's kind of sad lah. Not like very less people here, so I don't know whether they watch the video, they understand or not what is all this. Hopefully they do. Okay, so you have you have base substitution, base deletion, base insertion. Okay, and then examples. You have thalassemia, sickle cell anemia. Talking about sickle cell anemia. Siapa during exam, dia tanya sickle cell anemia, siapa pergi tulis loss of menstrual blood or lack of ferrum in diet? Dia ada 4 choices. One is loss of a lot of menstrual blood during menstruation. Second is lack of ferrum. Ferrum is iron lah, because Fe kan, Fe is iron lah, so ferrum lah, understand? Yes, we have that question in paper 1. It's okay if you don't remember. So you need speak lack of ferrum, okay. Or you didn't know what sickle cell anemia is, okay. Sickle cell anemia, okay, later I explain, you look at the textbook. If you choose those two, that means your answer is wrong already. Because those two, itu untuk anemia sahaja. Anemia maksudnya you tak cukup darah, you akan faint. Itu anemia, bukan sickle cell anemia. Sickle cell anemia itu satu gene mutation. Tapi anemia bukan satu mutation. Anemia is a condition lah. You loss of blood macam tu. Do you understand? Okay. Now you look at textbook. What is sickle cell anemia? Basically, the synthesis of hemoglobin, something go wrong already. So the shape akan crescent. Dia bukan, okay. Red blood cell, who can give me the proper term of the shape of red blood cell? Normal punya red blood cell. What is the shape? Dia ada satu special term. Biconcave, this shape. Yes, very good. This one very important. You must know. This is biconcave, this shape. Kenapa biconcave? Because, okay, concave is macam mana? Concave land. Masuk kan? Concave land go in macam tu. Concave. Okay? Biconcave, that means 2 sides go in. So you see your red blood cell, one, the atas sini akan masuk, bawah sini juga akan masuk sikit. That's why it's biconcave. This is circular. So dia akan biconcave this shape lah. Okay. Tapi sickle cell anemia, okay, sickle cell anemia, dia akan produce crescent shape. Kalau crescent shape, dia tak ada hemoglobin. Dia tak ada hemoglobin, dia tak ada heme group, dia tak boleh combine dengan oxygen to produce oxyhemoglobin. So you akan you tak cukup oxygen lah. Very dangerous lah. This is sickle cell anemia. Do you understand? It looks like a sickle ma. You see the crescent shape? It looks like a sickle. You know what's a sickle? The knife that looks like a crescent shape lah. Okay. Albinism. Cystic fibrosis. Let me search what is it. I forgot already. Cystic fibrosis is what. Okay, cystic fibrosis. Where's my phone? Okay, cystic fibrosis, inherited disorder, severe damage to lung, digestive system, and other organs lah. Because it affects the cell that produce mucus, which is goblet cell, and also the digestive juices lah. Okay, so itu cystic fibrosis. Okay. Thank you, Jana. Yeah. Okay, sickle cell anemia will die ma. Yeah, most probably will. Mutation, there's always a risk of death. There is always a risk of death, including albinism. There is always a risk of death lah. Okay, maybe albinism not so much, but sickle cell anemia, thalassemia, cystic fibrosis, there's always a risk of death. Okay. Okay, let me. Okay. Let me hear your applause. Oh, okay. I think that's all for the examples. Okay. Chromosomal mutation, kita ada 4 jenis. Kita ada deletion. Apa lagi Deletion, duplication, inversion. Inversion ni paling suka lah. And translocation. Okay, ni translocation jangan confuse dengan plants lah. Ni translocation not plants the punya apa phloem tu no Okay, so deletion, you look at the textbook, basically is one part of the segment delete lah, the B already ambil keluar. Duplication maksudnya you double it lah, the B dia duplicate lah. Okay, inversion maksudnya P arm tu tukar dengan Q arm. You tengok, the F is at the P arm, A is at the Q arm, tapi dia tukar dah. So A pergi P arm, F pergi Q arm. You understand? Translocation basically a non-homologous chromosome, I tukar the position, I tukar this one. All this is chromosomal mutation lah. It affects the chromosome. Kita tak tengok gene, kita tengok the chromosome. Do you understand? Can you understand? Yes, okay. This is the arm, right? Yes, the chromosome. Dia akan affect chromosome. Dia tak akan affect gene. Not to say tak akan affect gene lah, tapi it affects the entire chromosome. Okay, dia bukan lagi apa protein synthesis, transcription, translation, no. The entire chromosome. So, the examples of this, okay. Now, why, like I said here, dia tak akan 4-6 for chromosomal mutation because you see, dia akan ada berlaku satu mutation semasa cell division. You tengok. Non-disjunction. Non-disjunction maksudnya dia tak homologous chromosome ataupun sister chromatid fail to separate. You see, first possibility, kalau dia fail to separate during anaphase 1, homologous chromosome fail to separate during anaphase 1, you tengok macam mana I'm not sharing screen, no? I lupa dah. You all got looking at textbook, right? Okay. Oops. Kalau homologous chromosome fail to separate during anaphase 1, okay, so dia akan macam tu lah, okay. So the gametes will produce N+1, N+1, N-1, N-1. Okay. And then if they can separate at anaphase 1 but cannot separate at anaphase 2 for sister chromatids, they akan produce N+1, N-1. Okay. So this is called non-disjunction. Okay. Then you see what happens. Okay. Non-disjunction, kalau XX, let's say for example, satu gamete tiada kromosom. Satu gamete ada double chromosome. So, kalau fertilized by sperm, dia akan produce Turner syndrome female. Kalau fertilized by sperm, that is Y, zygote akan die. The zygote cannot exist if there's only one Y chromosome, dia tak ada X chromosome. Itu akan mati. Because like I said, Y is actually very weak one. You cannot survive on its own one. Okay, the X, you kena ada X. Okay, XO Turner syndrome, OY zygote die. Double chromosome, fertilized by Y sperm, Klinefelter syndrome male. Kalau female, fertilized by X chromosome, then you will have triple X syndrome female. Okay. Kalau spermatogenesis tak ada, XO Turner syndrome female. Kalau spermatogenesis double chromosome, Klinefelter syndrome female. Okay. This one you don't really have to memorize. You just have to see the diagram only. You understand? No. Don't have to memorize. See the diagram, you understand only. The ones you have to memorize, not to memorize also, you have to understand is the example. Okay. Listen very carefully. Chromosomal mutation confirm example dia ada syndrome. Syndrome punya word. Syndrome. You tengok ada syndrome, confirm you tak akan pilih gene mutation lah. You akan pilih chromosomal mutation. Syndrome. Okay? So, the first one we have is, what is it called already? Down syndrome, Turner syndrome, Klinefelter syndrome, Jacob syndrome, and Cri-du-Chat. Okay, Cri-du-Chat is like French like that lah. Okay, so Cri is crying, and then Chat is the cat. So it's cat cry syndrome lah. Okay, later we'll look in detail. What is it? Okay. So, okay. Now, Down syndrome and Cri-du-Chat syndrome, okay, is affecting the autosome. Autosome maksudnya yang 22 pair tu bukan sex chromosome. Klinefelter, Jacob dengan Turner akan affect sex chromosome. Down syndrome di mana? Down syndrome di 21. So Down syndrome kita panggil trisomy 21 because dia ada 3 21 kromosom 21. Okay. Cri-du-Chat, partial deletion of the short arm itu P arm lah, okay. Partial deletion of P arm of chromosome number 5, Cri-du-Chat. Okay. So you see the symptom cry is similar to a meowing cat lah. Okay. For Down syndrome, protruding tongue, flat nose, mentally retarded, slant eyes, all this lah. Okay. Understand? Klinefelter syndrome, Klinefelter will only affect males. Understand? XY, I tambah satu lagi X, double the X. Okay. So a sterile man, because of the X, dia macam girl lah. Dia sebenarnya boy, tapi because of the X, it looks like girl characteristic. So dia small testes, dia tak boleh produce sperm, possess voice and chest similar to a woman. Okay. Untuk Turner syndrome opposite, Turner syndrome is XO. Dia girl, tapi dia karakteristik macam boy. So, lack of secondary female characteristic, undeveloped breast and ovary, web-like neck and low IQ. Understand? Okay, and then Jacob syndrome is 47 chromosome, 44 plus XYY lah. So that means this one even worse lah. This is like so powerful, you know. Your sperm can produce double Y chromosome, you know. This mutation very powerful, you know. So male who is taller than normal and has problems with pimples on his face, slow in acquiring speech, learning disability, weak muscles which is hypotonia lah. So ini Jacob syndrome. Understand? Most importantly, yang paling terkenal is Down syndrome. Kalau tak, Klinefelter dengan Turner. All these three is very very popular lah. Jacob and Cri-du-Chat not that popular lah. Understand? Yes ah. That's why I say genetic sangat senang saja. Okay, so you look at this final, I think it's final. Yeah, final one, mutation in somatic cell. It involves a somatic cell, which is skin cell, eye cell, upper cell, except gamete lah. Mutation in gamete, that means your sperm and ovum lah. Mutation in somatic cell cannot be inherited by the next generation, but because your gamete is to reproduce offspring, you can be inherited by next generation lah. Disease is present only individual mutation, but for gamete, you can inherited by the descendant lah. Okay, so that's all for chapter 12. Now, the last chapter is also very easy and very short lah, genetic technology. Yes, lost CDS. The genotype for the sex chromosome must memorize. Genotype for sex chromosome. You mean like XYXX? Yeah, you have to memorize. Not to memorize, it's already like, must be in your brain like XY is boy, XX is female. Okay? Very easy only, XY male, XX female, like that. And also the no worries one lah, very easy only. And also the what is it called? Mutation punya chromosomal number. You kena ingat Down syndrome trisomy 21, so 47. Okay, Cri-du-Chat affect partial P arm of chromosome number 5. Okay, then you kena ingat Klinefelter syndrome itu 44 tambah XXY. Turner syndrome XO. Okay, Jacob syndrome XYY. Okay. Genetic engineering, you just have to read textbook lah. Okay. Basically, what does it mean is genetic engineering is a gene manipulation technique. I modify the gene, I modify the gene, the DNA to get what I want lah. Okay. So an organism that has already been produced by the recombinant DNA technology is known as GMO lah, genetically modified organism. And then biologists can recombine the DNA or genome to form an organism. Like, you know, Jurassic World, Jurassic Park, all those, they use all this genetic engineering to put, they extract the DNA from the mosquito, if I'm not mistaken. They extract the DNA, the dinosaur blood DNA from the mosquito, and then they recreate the dinosaur lah in the egg. Okay. Luckily, I ain't passing down my gene. Okay, GMO. So, okay, an organism that contains the recombinant DNA is known as transgenic organism. So, okay, we have a few examples here. GMO cow produces milk which does not contain beta lactoglobin, a type of protein that causes allergy among some children. GMO goat possess human genes with codes for blood clotting factor. So, the blood coagulation factor is found in goat milk. Purified, used to treat hemophilic patients. All this you just have to read and understand lah. Okay, GMF. For food, we call it GMF lah, genetically modified food. So here are the few how to say advantage and disadvantage. Advantage is of course resistance to herbicide, resistance to disease, application in medicine, tolerance to heavy metals, resistance to pests. So because you have high quality, you resistant to your disease, all this, so you won't have food shortages. You can prevent food shortages, reduce the cost, increase the nutritional value, reduce problems, reduce usage of pesticide, increase in the production because it's more resistant and also more reproductive lah. But the disadvantage, of course, endanger the natural species because everything is really artificial. You don't have natural already. Okay, and then also a possibility that the gene in GMF can affect you, can be transferred to human. For example, the antibiotic resistant gene. You eat too much all these manufactured GMF, then maybe you will have the disease or what may have adverse effect on human health and genetic material. Yeah. So Bacillus thuringiensis gene is inserted into corn to increase its resistance against insect pests. Okay. Super salmon is also another fish, and because of the genetic engineering, it becomes very big, grows at a faster rate. Potato, leaf in chloralkalimates, is very sensitive to low temperature and frost. Gene for Arabidopsis species plant enable the plant to tolerate freezing condition lah. Okay. Okay, insulin. Insulin is an important hormone which controls the blood glucose level. That means convert the glucose to glycogen lah. Okay. Last time, insulin has to extract from the pancreas of cattle or pigs to treat diabetes mellitus. But now we can sendiri produce the insulin already. So how do we produce? Okay, later we'll see how we produce. But beside that, we can also produce the hepatitis B vaccine, blood clotting factor, and growth hormone lah with genetic engineering. Okay, ini yang paling important. You kena faham the process macam mana. Okay, so the first thing, a spliced human DNA with insulin gene. Saya human DNA, saya ambil satu gene yang control the production of insulin in pancreas. Okay. Cut, confirm you cut with restriction enzyme. Okay, you akan cut dengan restriction enzyme. Okay. Lepas tu, your organism, your what is it called? Your bacteria. Okay. Plasmid. Okay, I draw the structure. Okay. Bacteria normally you bacteria and archaebacteria is prokaryote. Prokaryote means dia tak ada membrane bound organelle. Dia tak ada plasma membrane semua tu. So dia akan macam ni lah. Dia akan macam ni. Lepas tu maybe ada flagella lah. Okay, so dalam sini dia akan ada banyak macam ni. This is like the genetic material lah. But dia tak ada nucleus. Ingat dia tak ada nucleus because nucleus is membrane bound. So dia akan mungkin ada plasmid. Plasmid is also genetic material macam nucleus tapi dia floating macam tu. Dia floating macam tu, dia circular. Okay? Dia circular macam tu. So plasmid ini saya ambil, lepas tu saya cut dia. I cut the plasmid using the restriction enzyme. Okay? I cut this part using the restriction enzyme. Human punya gene, the gene in the DNA, I will do. Okay, DNA in human. Okay, akan ambil. Okay, the gene macam tu akan cut dengan restriction enzyme. Okay, so ini dengan ini, I tambah bersama. Okay, so Kalau saya mau tambah sama, ini gene saya substitute dalam ni, confirm when I want to join, I will use ligase. Ligase is an enzyme that I must use to combine anything. Gene lah or organelle lah, I must use ligase enzyme. Okay? Ligase enzyme to combine everything. So now, the plasmid contain the genetic material and the insulin gene, dia akan combine to produce the insulin. Tapi you kena transfer this plasmid into another bacteria. So I transfer it into here. This bacteria, okay. Okay, so sini. And then I letak the modified plasmid. I letak the modified plasmid dalam. So the bacteria, dia sudah ada genetic material dan and the necessary code to produce the insulin. Dia akan produce the insulin. Once it produce the insulin, dia because it will reproduce everything clone everything, okay, so akan banyak insulin lah. Banyak insulin, lepas tu the last thing is you purify it. Okay, understand? And that is how you extract the insulin. Can you all understand? Okay? So you don't have to extract the other insulin from other animals. You just take the gene and the DNA, how to say, like the genetic information to produce the necessary insulin enzyme. You masukkan ke dalam bacteria, the bacteria tahu macam mana produce insulin, lepas tu you purify. Okay, yes, very cool. That's why honestly I like biotech a lot. This is considered biotech lah. That means you manipulate the gene to produce something you want. Okay, then you purify lah, so you dapat banyak insulin lah. Okay, so what is biotechnology? Biotechnology is a field which utilizes technology or method to manipulate organism for production of biological product lah. Okay, that means let's say mungkin I mau produce satu benda yang apa lah? Okay, maybe like how to say? Ini example. Maybe I want to produce glucagon hormone, or I want to produce like just now the corn that is resistant against pests. All this lah. Yeah, basically it's quantum physics of bio. Yes, in a way you can put it like that. It's basically like a quantum physics of bio. Yeah, very fun lah. Okay, so application of biotechnology in life. Gene therapy is used to prevent genetic disease. So let's say your gene already got the mutation, so you akan cut the gene using restriction enzyme. You insert a normal gene, so using the virus. Okay, sini ya, you tengok virus, bukan semua virus bahaya tau. Virus is something like a vector. It's like it's not an organism because it's not a living thing. It's just like a vector that carries the necessary genetic information into your cell. Okay, so like COVID-19, it will infect your cell, and then to produce the necessary, dia akan insert the genetic material so that your body cells can replicate the COVID-19 virus. Understand? And this is how you kena jangkit lah. Okay, this is how you fall sick due to COVID-19 lah. Okay, so cell performs normal function. Okay, DNA profiling. This one also exam got keluar lah. So basically, if you identify criminal, you tengok lah DNA similar, not settle paternity and maternity dispute, genetic disease, sustainability of organ donor and recipient. Okay. Now, okay, this one you have to read lah, unfortunately. Okay, but if you watch CSI, then you know how the process lah. Okay, so basically, blood, semen, skin, anything that contains the genetic information or DNA is extracted, and then you cut the DNA into different sizes of DNA fragment, and then the DNA of different is then separated by gel electrophoresis. Okay, you separate the DNA fragment dulu dengan gel electrophoresis, and then you transfer onto the nylon membrane. If you should watch CSI, then you understand lah the process. And then radioactive probes macam X-ray, dia akan shine pada nylon membrane lah. Because you see, the DNA band, dia sudah is very thick, right? The band. That's why when the X-ray shine, dia akan black color lah macam tu. So the X-ray film is processed to show the position of DNA band that form the DNA profile. Understand? This is DNA profiling lah. I'm sure you all know how to profile, right? You all can do the exam question, right? Can you all do the DNA profiling punya exam question? Should be easy, right? Yeah. You just see lah, mana yang sama itu bapak dia lah. Yeah, I think it's number 2, yeah. You see lah, siapa sama, siapa sama. Okay, production of insect tolerant plants. Okay, BT cotton. Okay, so BT cotton is Bacillus thuringiensis lah. Okay, type of soil bacteria secretes toxin. Okay, so gene produces BT toxin from Bacillus thuringiensis inserted into plant genome. Pest feeds on the plant will produce BT toxin. The toxin affects the digestive system and kills the pest lah, but it won't kill the human lah. Okay, bioremediation, treat the oil spillage. You will use Alcanivorex borcumensis. Okay. Biotechnology and life. Okay, nothing much. This is just important. Agriculture, medicine, forensic, environment. Okay. Habis. That's all for our class today.